Hello, biology students. This is deer mouse fur color from the field to the beach. We're working on lesson 3.1, protein synthesis. In unit 2, we explored the process that produces eumelanin and pheomelanin, both which influence mouse fur color. Eumelanin, remember, is the darker color and pheomelanin is the lighter pigment. We discovered that there's a difference in the receptor protein between the dark brown and the light brown mice. This signal protein here doesn't attach in the lighter mice, and so it doesn't break up and can't release this comma-shaped signal protein right here to go to the cytoplasm and signal the cell to make melanin in the melanosomes. Well, the hormone can bind both types of receptor proteins in the extracellular space. In the light brown mice, the signal protein isn't activated. And this unit will explore why the hormone does not activate the signal protein in the light brown mice. So remember, this fits into a larger sort of context. Remember, the hawks were eating the light brown mice in the dark fields, like the woods. Basically, the light brown mice don't match in with the soil and the rotting leaves and the tree bark and stuff in the fields and woods. And the dark mice don't match in with the lighter sand. So the hawks kind of picked off the mice that don't match the environment. And now we're looking at why some mice are lighter. Why are these beach mice lighter? What's the mechanism inside the cells of the light mice that makes them lighter? And it has to do with this receptor protein that links to this hormone and the signal protein. And the signal protein, when it's broken open by this receptor protein, signals the cell to make eumelanin. If it doesn't work, the cell mostly makes pheomelanin, which is lighter. And then the mice fur is lighter. The receptor that sits in the membrane of the melanocyte, that's the specialized hair shaft cell that makes pigment or coloring for the hair, is a protein. What do you already know about how proteins are made? Write your ideas here and then discuss your ideas with a classmate. Well, you probably don't have a classmate handy, but I gave you a word bank here. You can certainly add your own words. Please write what you know and understand about protein synthesis in the box here. You can pause the video before you go to the next screen. Now it's our mission to try to figure out all the different steps of protein synthesis and put them in order. To understand where the receptor protein comes from and how the receptor proteins in the dark brown mice and the light brown mice are different, we need to understand how that information is stored in the DNA and is used to make a protein. We'll look at these cards. We're going to figure out what order they go in, and then we'll work on the next page. So you can see these cards have various different stages of protein synthesis on them. Transcription, kind of looking at the cell as a whole, the hormone, the finished protein in the cell membrane, the cytoplasm, where translation is occurring, the amino acids all lined up in the correct order to make a protein, the functional protein all folded correctly. We can look inside the cell, kind of looking at those specific organelles that do particular jobs in the cell, and then also looking deeper inside the nucleus to the DNA. So let's think about how those different steps, what order they go in, and just kind of refresh our memory a little bit about how protein synthesis works. So in order to do this exercise, you're going to need the document called Lab 1.1, Deer Mouse Lesson 3 Tables. So look for that in Schoology, Deer Mouse Lesson 3 Tables. It's got some information on protein synthesis that's going to help make sense. And then it has a table that you need to fill out with steps 1 through 8. You'll put the letter of the card that's step 1, the letter of the card that's step 2, the letter of the card that's step 3, and then you'll explain what's going on in that picture or that card. So now that you've got your table downloaded, that's lesson 3.1, table 1. It's in Schoology under the main lab assignment. We can go look at our cards. I've lined them up here on a Jamboard in order. We're looking at the large cell first, kind of the overview of the whole process inside the cell. Then we're looking at the nucleus, kind of focusing in on a particular organelle of the cell, looking at the DNA in that nucleus, so that comes next. Then we use the DNA to make RNA. That process, remember, is called transcription. After transcription, we do what's called translation. We use that RNA template that we made up in transcription over here. Here's transcription. We use that RNA that we made from DNA, and we go to the ribosome inside the cytoplasm, and we use tRNA to bring in amino acids that match that 3-letter codon on the mRNA. And you can see the tRNA right here. It's bringing in an amino acid, it's connecting them up, and eventually you get a string of amino acids in the correct order that make a functional receptor protein here. Remember, only if you get the right amino acids in the correct order will the protein fold and be functional. So here is a functional protein that's in the dark mice, and you can see that functional protein in the cell membrane here doing its job. Working with hormones in the extracellular space, that hormone hits the receptor protein then takes that cue from the hormone and breaks the signal protein in half. This half, the sort of comma, turquoise comma, breaks off and goes to the cytoplasm of the cell to create and signals it to create eumelanin, which is the darker pigment. So that's kind of a summary of the protein synthesis process, and you can see what the order is from left to right. B, G, H, A, D, F, E, C. Now that we've filled out that protein synthesis table, let's go to the next page here. Now, we made a model earlier in this lab. We made it in the lesson 1.1 and 1.2. We're going to upgrade our model with new information from Unit 3. You can start with your model from Unit 1 that you added to in Unit 2, or you can just do a simplified condensed version of your Unit 2 model and add Unit 3 information. I will show you a version of a model. It's made in what's called Sage Modeler, which unfortunately does not work on the iPad. You are welcome to make yours on paper and just take a picture and upload that image. You can also do it on an art app. You can do it in a Jamboard. You could do it in Google Draw. You've got lots of options for how you want to do this model. You just need to upload an image when you're done. So here's a bit of a simplified version of the model. I have kind of the whole cascade of the different biochemical things that are going on in the cell membrane, the hormone signaling the cell with the receptor proteins breaking up those signal proteins, the activated signal protein signaling the cell to make eumelanin, which is great if you're a mouse in the field habitat. I have some of the transcription and translation information here from protein synthesis, how we make RNA in the cell's nucleus, we translate that in the ribosome, we line up amino acids, and we make a functional protein. Now, if we have a mutated protein, we're mostly going to make pheomelanin, which is great if you're a beach mouse. Otherwise, if you're making a dark protein on the beach, you're going to get eaten by hawks. This is a simplified protein, simplified model. It's not the correct answer. Don't copy it, but you get the idea of what to do here. You need to tell me why the light mouse and the dark mouse are different and why that's important. So what biochemical, what cell process, what's going on in the cells of the mice that make them look light and dark. And why does that matter when they live on the beach or the field habitat? Make sure your model reflects that there might be a protein mutation that makes the mice different, that makes them survive in different habitats better or worse. For example, that light mouse with the mutation might not survive well in the field habitat because it's a darker habitat, where the field mice might not survive well on the beach because they're too dark and the hawks are going to see them easily. Remember, when you're uploading your model, the image of your model, next in the text box next to your model, you need to write a detailed explanation of how your new model is more inclusive of what you learned about and better represents how mice get their fur colors. So don't just draw pictures. Tell me about the model. Tell me about why, at the cellular level, what's going on in the mice's cells, particularly their cell membranes and that receptor protein that's making them different colors. Consider the process of protein synthesis and the fact that the receptor proteins in the dark brown and light brown mice function differently. Make a prediction about what causes this function. Well, we know quite a bit about what causes this function. We know that something is going on with that receptor protein that causes it to function differently. If you need a little bit of review here, this is lesson 2.2, the compare. It's comparing light and dark cells. And here is the dark cell. You can see that the signal proteins are attaching, they're breaking in half, and then sort of the turquoise comma looking activated signal protein is going into the cell cytoplasm and making this eumelanin. So if we look at the cytoplasm when we measured it, it has signal protein. It's making this eumelanin, the darker melanin, and it's got this hormone. If we look at this, the lighter mouse here, that signal protein just isn't attaching at all. And so it can't break apart, and we don't see any activated signal protein here like we do in the dark mouse. So the activated signal protein is different. We're also not seeing any of that darker eumelanin in the melanosomes, those organelles that are specialized for making melanin in the melanocytes. So if you're a little confused, go back to lesson 2.2 and go to the lesson called, the part of the lesson called Comparing Light and Dark Cells, and that'll give you some ideas about how to write your prediction for lesson 3.1. So again, if you're kind of struggling with making a prediction, go back to lesson 2.2 where you can compare light and dark mouse cells at the cell membrane level and check that out. So now it's time to click Show All Answers. You want to go to this little triangle at the very top of your device, and then you want to click click this square. You're going to click Options on top of the menu that pops up. It's not the same on my laptop. You're going to click Options, then you're going to go PDF, then Return, and click Notability. So click the box, click Options in the menu, click PDF, and then go back to Notability and download it to Notability. And from there, you can upload it. So this is lesson 3.1 in deer mice protein synthesis. The next video will be lesson 3.2. See you next time. This is Dr. B signing off.